Hey, so obviously I'm not in class today, so I'm pre-recording your lecture. Uh, Last time we met, we discussed the process of transcription. So just to remind you where we are in the chapters and in the syllabus, we've been doing basically what's called gene expression. This comes from section 2.7, 7.2, and 7.3. Gene expression is taking the instructions in your DNA, using those instructions to make some proteins. And we talked about transcription. We discussed how transcription gives you an immature mRNA molecule, which then has to get modified. So if you look back at your notes, you should see information about RNA modification, including the process of adding a methyl cap to the 5' end, poly A tail to the 3' end, and then splicing, which includes SNRPs, best vocab term of HL Bio, SNRPs, spliceosomes, which then help to remove introns, connect exons, and give you a fully modified, mature mRNA molecule. After transcription's done, after post-transcriptional modification is done, that mRNA molecule leaves the nucleus, goes out to a ribosome for protein synthesis. And that is what we pick up today, translation. How we go from mRNA to actual synthesis of proteins. So let's get started. So actually this slide is from last time, but I want to start here just to remind you where we were. So everything from last lesson, transcription, was occurring in the nucleus. This is the nucleus here. And when transcription is done and post-transcriptional modifications aren't finished, then the mRNA molecule is going to be guided through one of these little holes. And there's a zoomed in version of one of those holes. So this is the membrane of the nucleus here. And in the membrane, there are these little pores. And so the 5' end has that methyl cap, which helps to guide the mRNA molecule out through one of these pores. And then it can go out to a ribosome. And that's where translation starts. So for translation, here's the translation. We're going to add in a structure called a ribosome. You guys know what ribosomes are. They build proteins. The hang up here, the reason that translation is a bit more complicated is that, remember, with transcription, we were taking DNA and copying it into RNA. We were taking DNA nucleotides and copying them into RNA nucleotides. But at the end, nucleotides are all pretty similar. You've got your sugar, your phosphate, and your base. So the nucleotides were pretty similar, so that RNA polymerase enzyme had a pretty easy time copying that gene. When we go to translation, on the other hand, now we're taking RNA and we're using those instructions to build a protein out of amino acids. And amino acids are crazy. There's 20 different types of amino acids, and they're all very different. And so linking amino acids together in the right sequence to make a protein is going to be more complicated. And so these steps are going to be a little bit trickier today. So let's take a second and really explore what's happening in this GIF, and then I'll start to get some nice things for you to take down in your notes as we go forward. So, ready? First vocab term. What we're looking at here, well, first of all, you should probably note that translation occurs in the ribosome. Important thing to jot down. So go ahead, write it in. Translation is occurring in the ribosome. And your mRNA molecule is being fed through the ribosome and being read 3 letters at a time. So, what are we looking at here? Here's your first new vocab term. You had it in 9th grade, but as a reminder, these little guys that are floating in and then floating out, these are very specialized types of RNA. It's another type of RNA. It is called tRNA. I'm using my pen here. We got T, R, clicky, 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 tRNA. That T stands for transfer. Transfer RNA, tRNA. And you can see what it's doing here. These little RNA molecules are transferring the correct amino acid toward the ribosome. And so over here on the left side of the screen, you can see this is a tRNA molecule without an amino acid attached to it because it's done its job. And now it's going to go back off somewhere into the cell and pick up another amino acid. Over on the right, these are the tRNA molecules transferring the next amino acid, which is going to get added to this chain up top. And this chain up here is basically a long chain of amino acids. And when it's done, it's going to fold together into a protein, because proteins are made of amino acids. And so these molecules, these tRNA molecules, are transferring the correct amino acid at the correct time, which will correspond to the code that you will see down below on the mRNA. So, tRNA molecules transfer amino acids to the ribosome to be built into a protein. Now, the reason we have to do all of this extra building with this extra equipment of the ribosome is that, remember, these amino acids, they're all different, and some of them are quite large, some are quite small, and so it's not quite as simple as just taking an enzyme and then just copying a bunch of nucleotides. So we need to be a bit more careful here. Here's how it works. The mRNA molecule gets bumped down 3 letters at a time. 3 letters at a time. And if you look closely, you'll see that there are these little 3-letter sequences, which then are complementary to sequences on the tRNA. Check it out. AUG just came in, and it's complementary. Here's, oh, sorry, it's a GIF, so it keeps recycling. Here comes AUG. It's complementary to this UAC on the bottom here. So there's going to be a match between what's on the mRNA and what's on the tRNA. And what happens is the ribosome checks to see that that match is correct. If that match is correct, the amino acid gets linked in, and we can keep going. If it's not, then this tRNA molecule is basically going to get kicked out and until the right one actually shows up. Okay, so here come the notes. Ribosomes, during translation, will work together with T or transfer RNAs to basically assemble a polypeptide. That's the structure of the protein. So right here we're looking at a ribosome, and the ribosome is made up of 2 basic units. There's the large subunit on top, the small subunit on the bottom. Those guys clamp together, and then your mRNA molecule gets fed through there. As the mRNA molecule is read, tRNAs bring in the right amino acids, and you can build a polypeptide. So ribosomes and tRNA assemble polypeptide chains from the mRNA instructions, the mRNA transcript, as it were. And like I was saying a second ago, the mRNA molecule is going to be read into little words. These are 3-letter words, which we call triplets or codons. Codon is the preferable term. I almost always see the word codon. Sometimes you'll see this word triplet too, because, IB sucks. But we'll just deal with both. So your mRNA is read 3 letters at a time, and those 3-letter sequences are called codons or triplets. So in red, in this diagram, the red strand, that's the mRNA. And the codon in this example is UCG. Now, what's happening is that while that sequence is locked into the ribosome, the tRNAs are going to be floating around, and only the one with the correct anticodon, or the complementary sequence, is going to be locked into the ribosome to actually connect an amino acid. And that complementary sequence on the tRNA, what we're looking at right here in green, how festive, this is the anticodon, and it's going to be complementary to what's on the mRNA codon. So again, this red stuff, this is the mRNA getting fed through the ribosome. This is the sequence, the anticodon, that just was floating in from that tRNA. So our tRNA has the anticodon, the mRNA has the codon. And if there's a match, then the amino acid up at the top of this guy gets attached. So first, before we go farther with the processes of translation, let's look at the structure of a typical tRNA molecule. You will need to know or recognize this structure. So I recommend that you just make a very quick sketch of this in your notes. So if you're, it's RNA. It's just like any other RNA molecule. It has a 3' end, which then goes all the way around to the 5' end over here. But these tRNA strands are uniquely designed so that they maintain a consistent shape, and they only serve one function, which is to basically bind to an amino acid and then carry that amino acid to a ribosome so that it can be added to a protein. And then it goes back and it finds another amino acid and does it all over again. So what you need to know about the structure of a tRNA molecule is that up at the top here, where the 3' end is up here, the top right there where it's kind of opened up, there's a place where the amino acid will bind. It's called the amino acid accepting end. Nifty. Then down below that on the opposite side, down here, this is where the anticodon is located. This is the 3-letter sequence, which must be complementary to the codon on the mRNA to check for a match. So that's the structure of the tRNA molecule. You'll need to know that basically, if you see a diagram, you should know where the amino acid would go and where the anticodon would be. Okay, and this is, you might not like this diagram, and I'm sorry, but we'll see what we can do. Here's how it works. Basically, you're gonna wind up with two. I know. Let me go back again. These little loops 3 and loop 1, they're not included on this diagram just to simplify things. But you have your amino acid accepting site up here. Here's the anticodon down there. And so right here, this in the middle here, this tRNA molecule has been accepted. It was not rejected. There was a match. You know, the anticodon matched the codon. So then it moved in here to the second site. And it basically has already joined its amino acid up at the top. And now, the next amino acid is coming in. The next tRNA molecule has just arrived right here, and it has the anticodon UCG. So the ribosome at this moment is checking to see if there's a match down below, to see if there's a match here with this codon, AGC. And yes, these are complementary, so yes, there's a match. And so what's going to happen is that while these two guys are still stuck in the ribosome, they're going to form a special bond between amino acids, which you already know the name of, but maybe you forgot. They're going to form a peptide bond. So this peptide bond, this glowing yellow bond, this peptide bond is forming in between these two amino acids. And then once they're linked together, then this tRNA molecule can detach and move on its merry way, just like this last one has just done. And then this process keeps going over and over and over until you get to the end of the gene. And as it's continuing, these polypeptide chains just keeps on growing. So in the ribosome, we have 3 sites, if you will, or 3 zones. And these 3 sites are called the A site, the P site, and the E site. Think of A for acceptor, P for peptide, and E for exit. So acceptor. At the A site, at the acceptor site, the ribosome is checking for a match between the codon and the anticodon. I'll say it again. At the acceptor site, the ribosomes are checking for a codon-anticodon match. If there's a match, we move on to the P site, P for peptide. So if there's a match between the anticodon and the codon, we move on to the P site where the peptide bonds will form, P for peptide. After the peptide bonds have formed, we move on to the E site right here, and that just stands for exit. That's where we're going to kick the empty tRNA molecule. We're going to kick it out, APE. And now we're back at this GIF again. Take a second and just watch it go. See if you can, with the notes you have, looking at this, does it make sense? Can you explain it to yourself? Can you explain it to your parents? Can you explain it to your dog? Okay. Onward. Now, at the beginning of every gene, there basically is a sequence, a 3-letter sequence, that always starts off a protein. Now, this stuff at the front, closer to the 5' end, we don't know what this is. It could be anything. But the important part is that whatever these letters are right here, these they could be U's or A's or C's or G's, we don't know. They don't have the sequence AUG. Until we get to that AUG codon, the protein is not going to start. It's a signal to basically start the translation process. And so every gene has to start with the same codon, which is AUG. It's always the same start. And that gives us, it always gives us the same amino acid, which is methionine, MET. So that's always the start of a gene. And then once that gets going, we continue with translation until we get to the end. And how do we know we're at the end? Well, we don't go all the way to the end of the sequence. See if you can think about why. On the 3' end, what is on the 3' end of this mRNA molecule? I know you can do it. Think about it. Think about it. Think about it. Pause the video if you have to. Okay, I'm going to give the answer now. Spoiler alert. On the 3' end, you've got a whole bunch of A's. The poly A tail is there. And that's not going to be very useful for your proteins. And so we don't want that A nonsense to be added to your proteins. So we always start with methionine, and then when we get close to that poly A tail, there's always going to be a codon that says basically stop. Stop what you're doing. We're done. It's a sum sequence that basically, it's 3 letters, and instead of giving us another amino acid, it basically just derails the whole process, the ribosome splits apart, and we're done. We're not going to keep doing any more translation on this particular gene, and that's the end. So there's always a start codon and a stop codon. The start codon is always the same, and it always gives us methionine, and there are a few different ways to stop it. There are a few different sequences which will stop translation. And we'll talk about that next class. Okay, the last big concept for today is strictly an HL concept. It's not hard, it's just extra stuff. But what you have to understand about translation is that we're not just talking about one ribosome working on one, my hand's backwards. We're not talking about one ribosome working on one mRNA molecule. In fact, there usually are multiple ribosomes all working at once. And if you look in this photograph, this is a micrograph, so a photograph taken with an electron microscope. If you look at this electron micrograph, what we're looking at here is, it's kind of hard to tell, but if you look closely, I'm going to start on the right. Going from right to left, there's kind of a faint line that's connecting all of these beads. And that faint line is our mRNA molecule. And then we have a glob and 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 all these little beads, if you will, on the necklace. Each of those globs, those are all ribosomes. So there's a bunch of ribosomes all working together on this same mRNA molecule. And each one of them is basically translating the same gene. And so what we can do is we can regulate the rate of gene expression by changing the number of ribosomes present on each mRNA strand. So your big vocab term here is this word up on the top right, a polysome. Not a hard concept. Here's what a polysome is. A polysome is basically a collection of multiple ribosomes all working on this same mRNA strand at the same time. Right? So a polysome is just what we're looking at. This is a picture of a polysome. It's a bunch of ribosomes all working on the same mRNA strand or the same gene at the same time. Now, why do you do that? Because then we can make a whole bunch of copies. So let's say it's a cell in your digestive tract, and it needs to basically, no, that's a bad example. It's a cell in your pancreas, which is making enzymes for your intestines. And that cell in your pancreas needs to just make a crap ton of enzymes so that you can digest your food. And the way it does that is by utilizing these polysomes so that you can make many, many copies of that same enzyme. Instead of just one at a time, you can make a whole bunch of enzymes all at once with a polysome, because each one of these ribosomes is going to be making a protein. So it helps to increase the rate of gene expression. Now, pause for a second. We can regulate the rate of transcription through methylation and nucleosome spacing. Remember that? So we can regulate the rate of gene expression there. We can alter the type of genes which get expressed by splicing the mRNA molecules to make slightly different varieties using SNRPs and spliceosomes. Remember that? And now third, we can alter the rate of gene expression still here in translation because the number of ribosomes might change or the number of polysomes might change. So we can basically increase the rate of gene expression by having more ribosomes present, or we can decrease the rate of gene expression by having fewer ribosomes present. So polysomes help to regulate gene expression during translation, and that's why it matters. Okay. Kind of to wrap things up, the sequence of codons on the mRNA will determine the sequence of amino acids in the polypeptide, which determines the shape and the function of the protein. And so this is how nucleic acids function as the genetic code, which then give instructions, and the way we look and the way we act and the way we think. All of that comes down to the amino acids in the proteins, which come down to the instructions from the DNA. So this comes back to the central dogma of molecular biology, which is sort of the idea that DNA makes RNA, which then gives us proteins. And there's this sort of idea that we have one gene, which then gives us one polypeptide, which is, of course, a little bit off, because, like we said last time, with splicing. Okay, this link, you should follow this link. It's a really nice visual of everything we just did. It's the same people that made the transcription video that I showed you, same people that made the DNA replication video that we watched before. This one is probably the most clear of them all, and I think it really helps to give you kind of some clarity on everything that I was just talking about. I feel like I could describe this to you for hours, and it wouldn't be as good as just watching a video with the animation going. So watch this animation, then go back and reflect on your notes, and make sure you understand everything you have down. And then, when you're done, well, you can't do this at school. When you get home, make sure you read these sections in the textbook, and then the next time we meet, I'll give you some sample questions. We'll make sure that it all stuck, and we'll kind of go back through it real quick. But rewatch any of this as needed. But this is your lecture on translation. We'll go into some sample problems and questions and talk about the details of translation next time. But this is your foundation, so please make sure you get it, and then prepare questions for me for next class for anything that did not make sense in this video. And good luck. I hope to see you soon. And that's it. Ta-ta.